Today, we're diving into a massive biological mystery, one that's hidden inside every single one of your cells. It's a story about a huge scientific mistake that lasted for decades and the incredible journey to finally set the record straight. 98%. Just let that sink in for a second. That's how much of our DNA does not code for proteins. And you know, proteins, they're the rock stars of biology, the building blocks that make up, well, everything. Our muscles, our enzymes, the works. For the longest time, we were only focused on the tiny 2% of our DNA that actually makes them. So, what did we call that other 98%? You're not going to believe it. For decades, it had this simple, catchy, and honestly kind of insulting name, junk DNA. Yep. The thinking, starting way back in the 70s, was that all that extra stuff was just evolutionary garbage, useless filler that was just along for the ride. But, I mean, does that really make sense? Could our biology really be that inefficient? Carrying around all this junk? It just didn't sit right with some scientists. And that brings us right to the heart of what we're talking about today. What if the junk isn't junk at all? What if we've been completely wrong about almost our entire genetic blueprint? Okay, so let's dig into how this whole idea, this scientific dogma, started to crumble. Because even when everyone was saying it's junk, a few brilliant minds were looking at the evidence and thinking, I'm not so sure. It's a classic story of how science really works, by questioning everything. You can really see the whole story unfold on this timeline. I mean, look at this. Way back in the 1950s, Barbara McClintock was already proposing these controlling elements. But nope, the junk idea took over in the 70s anyway. It wasn't until the Human Genome Project in 2003 that we really saw the scale of the mystery. And then, bam, 2012, the ENCODE project drops a bombshell. This so-called junk DNA was humming with activity. And we have to talk about Barbara McClintock for a second. She was so far ahead of her time, it's incredible. Her idea of controlling elements was mostly ignored at first, but her work, which eventually won a Nobel Prize, by the way, showed that the genome wasn't some static library of books. No, it was dynamic. Things were moving around, jumping from place to place, controlling other genes. It just took the rest of the world a few decades to catch up. All right, so if it isn't junk, then what in the world is it? Let's take a little tour of what scientists now call the dark genome. It's a great name, right? So what's actually in there? Well, it's not just one thing. It's more like a whole complex ecosystem of different types of DNA sequences. Check this out. The biggest piece of the pie, almost half, is something called transposable elements. Then you've got introns, which are these bits inside genes that get snipped out. That's another quarter. And then, get this, endogenous retroviruses, actual ancient viral DNA, make up a solid 8% of who you are. It's just a wild and fascinating mix. So those transposable elements, the biggest chunk we just saw, they have an awesome nickname, jumping genes. And it's for a good reason. They can literally move around, cutting themselves out of one spot and pasting themselves into another. And you can imagine, that ability to just up and move gives them a massive potential to shake things up across the entire genome. Okay, this next part is my absolute favorite. A huge part of your DNA is literally made of ancient viruses. I'm not kidding. They're like the fossilized remains of infections that happened millions of years ago to our ancestors. That viral DNA just wove itself right into our genetic code and has been passed down generation after generation. It's just mind-blowing, right? A living record of our evolutionary past. Okay, so we've seen what this stuff is. Now for the really big question. What does it all do? Because what we're finding out is that this dark genome isn't some random collection of genetic weirdos. It's more like a huge, complex, hidden control panel that's secretly running the whole show. This quote really just nails it. The Human Genome Project, it gave us all the letters of our genetic code, right? The A's, T's, C's, and G's. But it didn't tell us anything about the grammar. And that's what this non-coding DNA is. It's the punctuation, the chapter breaks, the instructions that tell the cell how to actually read the genetic book. Without the grammar, the letters are just gibberish. So what's its main job? Gene regulation. The best way to think about it is like a massive, complex control panel covered in switches. Every single cell in your body has the exact same DNA blueprint. So why is a brain cell so different from a skin cell? It's because different sets of these switches are flipped on or off. And those switches, they're all hidden away in that dark genome. And it gets even cooler. It's not just the DNA sitting there. A lot of it gets copied into something called non-coding RNA. And these little molecules are the real workhorses. Some of them are like matchmakers, bringing different proteins together so they can work as a team. Others are like a cellular GPS, with little zip codes that tell proteins exactly where to go. And yeah, some are just straight-up on-off switches, controlling gene activity directly. And we're not talking about just a few of these things. Scientists have found over 25,000 different types of these non-coding RNAs so far. 25,000! And the craziest part? We only have a clue what a tiny, tiny fraction of them actually do. It's like we've just discovered a whole new, incredibly complex layer of our own biology. So, why should you care about all this? What does it actually mean for us in our daily lives? Well, it matters a lot, because this is completely changing how we understand human health and disease. This isn't just some abstract science lesson. This is absolutely critical for the future of medicine. Get ready for this number. 90%. Studies are finding that about 90% of the genetic links to common diseases, they're not in the genes themselves. They're in these non-coding regulatory regions. They're in the dark genome. So think about what that means. For a whole host of diseases, the problem isn't a broken gene. The gene is perfectly fine. The problem is with the switch that controls it. A tiny mistake in that dark genome can flick a gene on when it should be off, or turn it way down when it should be on full blast. It's a total shift in how we think about where disease comes from. And we're talking about some of the biggest health challenges we face. Cancer, heart disease, Alzheimer's, autism, autoimmune disorders, the list goes on and on. All of them have now been linked to this regulatory DNA. So if we want to tackle these conditions, we have to understand the dark genome.
This is a genuine paradigm shift. I mean, the old way of thinking was simple. Bad gene, bad disease. All the problems were in that 2%. The new view? It's much more subtle. Disease is often caused by a problem in the 98% that's controlling the genes. It's just a completely different ballgame.
So what's next? Well, we are standing at the edge of a brand new frontier in genetics. Seriously, exploring our own biology has this vast new territory. And honestly, the real work is just beginning. And believe me, even with everything we've found, we are just scratching the tip of the iceberg. We thought it thousands of these little switches and RNA molecules, but for most of them, we still have no idea what they do. It's like having a map of the world where 98% of it is just labeled, "Here be dragons." There's so much left to explore. But the good news is, we have some incredible tools for this expedition. Things like CRISPR gene editing let scientists go in and literally flip these switches on and off just to see what happens. And we're using AI and deep learning to sift through mountains of data, trying to predict what all these mysterious sequences might be doing. It is an absolutely amazing time to be in this field. You know, the whole junk DNA story is just such a great reminder of how science actually works. It's not about having all the answers. It's about questioning what we think we know. It shows us that even our most basic beliefs can be completely upended by new evidence. And it leaves you with a really big question to chew on. If we were this wrong about 98% of our own instruction manual, what else about ourselves have we gotten wrong?